So a peer-reviewed journal is basically an article that a scientist has written, and what they do is that they send it to a journal editor, and then the journal editor sends it to three other scientists that are experts on your field. And so what they do is that they read your paper in order to determine whether your paper is good enough to be published. So typically, there's three different things that can happen with your article. So the first thing is that they're gonna say, wow, this is a really good article, let's just publish it as it is. Or the second, that is the most common one, is it's good, but we have some questions. And so what they do is they send you those questions and you have to address their concern. Or the third thing that could happen, but it occasionally happens, is that they tell you, oh no, this is not a good study at all. So don't even bother to send it to our journal. And the beautiful part about the peer review process is that it ensures that only the best data is published. So after reading scientific journals or after doing a little bit of research, then you can use that information to design a hypothesis. Now, a hypothesis needs to have two very important components. A hypothesis must be testable and must also be falsifiable. And in a minute, we'll talk about what do we mean by testable and falsifiable. So in this case, we have refined our hypothesis. So now the hypothesis, caffeinated coffee improves alertness. So a hypothesis must have two very important components. And it must be testable and it must be falsifiable. So what do we mean by testable? It means that you must be able to test it somehow. You must be able to design a careful experiment in order to test that question or that hypothesis. Falsifiable means that once you collect your data, and once you analyze your data, then either the data is going to support or reject your hypothesis. In science, we never say that our hypothesis is correct. And the reason for that is because if you remember, science is always evolving. We're always coming out with new ways to test our hypothesis. And what was true 10 years ago, it might not be true today. Again, in science, we never say that our hypothesis is correct. We can only say that our data supports our hypothesis. Let me give you some examples of hypotheses. So in this case, if I replace the battery in my car, then my car will get better gas mileage. So is this hypothesis testable? And is this hypothesis falsifiable? Yes, all I have to do is just replace the battery in my car to determine if I get better gas mileage. If I add fertilizer to my garden, then my plants will grow faster. Is it testable? Is it falsifiable? Yes. All I have to do is to add fertilizer to my garden to see if the plants will grow faster or not. As I mentioned before, in science, we never say that a hypothesis is correct. And the reason for it is because science is always evolving. We're always coming out with better microscopes or new technologies to test our hypothesis. And what was true 10 years ago, it might not be true today. If our data rejects our hypothesis, then that hypothesis is no longer going to be considered as an explanation for a phenomenon that we're studying. If, however, the data supports our hypothesis, then that hypothesis is going to be considered as an explanation for the phenomenon that we're testing until someone tests our hypothesis later on, and maybe at that point the data is going to reject that hypothesis. Okay, so now that we have our hypothesis, then the next thing is to test our hypothesis. And we do this by designing an experiment. Now, it is extremely important that when we're designing our experiment, that we do it very carefully. So we must have at least one experimental group and at least one control group. So does that mean that we can have multiple experimental groups? Of course. And we can also have multiple control groups as well. But we'll talk more about those in a minute. So after you carry your experiments, the results are either going to support or reject your hypothesis. All right. So in this particular example, what we're trying to do is to measure the effects of drinking coffee. So what happens after a group of individuals drink caffeinated coffee? So what happens to them in terms of concentration? What happens to them in terms of energy? So that's basically the experiment that we're trying to test here. All right, as I mentioned before, we must have at least one experimental group and one control group. So the experimental group, it's the group of participants who actually get the new treatment. So those are the ones that get the new manipulation. So in this particular case, are going to be the individuals that are drinking caffeinated coffee. Okay. So another example will be if we are trying to test a new treatment for a specific disease, the experimental group are the ones that are going to get the new treatment. Now you must also have a control group. So a control group gets something that looks similar to what the experimental group, but is not supposed to have any effect. So in this particular case, we'll be thinking about drinking decaffeinated coffee. Okay. So on another example would be if we're testing a new drug, the control group are gonna get what we call the placebo. So the placebo is basically a pill. A lot of times they call it a sugar pill that looks like what the experimental group is taking, but it's not supposed to have any effect on those groups of individuals. And the reason why the control group must also get something instead of nothing is because we want to keep them blind, essentially. If the control group knows that they're not getting the new treatment, then what they can do is they can actually out of the experiment, or that can have an effect on our results. All right, so the experimental group gets what we call the independent variable. So in the independent variable, it's the factor that's been deliberately changed in the experiment. Okay, so in this particular example, we'll be drinking caffeinated coffee, and as I mentioned before, you can have multiple experimental groups. An example of this will be if you're trying to test different concentration of a drug to determine what concentration is more safe to give to the individual. So you're going to have different experimental groups in that case. Now, that control group, right? So that's the group that is not going to get like the new treatment. Okay. So rather than not giving them anything, right? So we want to keep the control group as similar as to the experimental group. So the control group receives what we call a placebo. Another term that we use for the placebo is the sugar pill. So they are getting something, but it's not supposed to have an effect on the control group. Okay? And so the control group serves as a way to compare the experimental group to see if the new treatment is actually working or not. Okay, so the dependent variable is the measure of the results in an experiment. So essentially the dependent variable is equal to the result of the experiment. And on a graph, the dependent variable always goes on the y-axis. Okay, so let me say this one more time. The dependent variable is the result of the experiment. And on a graph, they always go on the y-axis.